Today, Group 21 is presenting Cystathionine gamma Lyase deficiency.
For the enzymatic reaction, cystathionase uses pyridoxal phosphate to facilitate the cleavage of the sulfur gamma carbon bond of cystathionine, resulting in the release of cysteine. The external ketamine is then hydrolyzed, causing the release of alpha-ketobutyrate. The lysine residue reforms the internal aldehyde by kicking off the ammonia-leaving group, and the amino group on cystathionine deprotonates and undergoes a nucleophilic attack of the internal aldehyde. An additional deprotonation by a base results in the formation of the external aldehyde and removal of the lysine residue. The basic lysine residue is then able to deprotonate the alpha carbon, which then pushes the electron density into the nitrogen of the pyridine ring. An alpha-beta unsaturation occurs by this and pushes a lone pair onto the aldimine nitrogen, which then cleaves the sulfur gamma carbon bond, resulting in the release of cysteine. A pyridoxamine derivative of vinyl glycoxylate remains after the gamma elimination. The lone pair from the pyridine nitrogen pushes electron density to the gamma carbon, which is protonated by lysine. Lysine then attacks the external aldehyde, pushing electron density to the beta carbon. The imine is then hydrolyzed to release alpha-ketobutyrate. Deprotonation of the lysine residue causes ammonia to leave, which then completes the catalytic cycle.
The CTH gene encodes for the enzyme cystathionase, or cystathionine gamma Lyase, and it is located on chromosome 1. The enzyme usually breaks cystathionine into cysteine. However, a mutation in the CTH gene weakens the enzyme's affinity for pyridoxal phosphate, or PLP. PLP is a prosthetic group necessary for the function of this enzyme. Because the mutation causes enzymatic activity to decrease, an abnormal amount of cystathionine accumulates and leads to increased urinary excretion. This mutation is inherited in an autosomal recessive pattern, meaning that one copy of the defective gene must be passed down from each parent in order for the child to show the disorder. A study published in 2003 revealed 2 nonsense mutations and 2 missense mutations in the CTH gene were associated with cystathionine urea. Research is still being done to conclude specifically which mutation causes this deficiency.
Normally, this enzyme has a role in regulating cell growth and making hydrogen sulfide. This hydrogen sulfide enhances the outward flux of potassium by opening potassium channels, thus leading to hyperpolarization of membrane potential and smooth muscle relaxation. So, without this enzyme, it results in hypertension because the smooth muscle can no longer relax. Additionally, pregnant mothers with this deficiency have pregnancy complications such as proteinuria and thrombocytopenia. And what these two are is proteinuria is the presence of abnormal proteins in the urine, which may indicate damage to the kidneys. And thrombocytopenia is a deficiency of platelets in the blood. And this causes bleeding into tissues, bruising, and slow blood clotting after injury.
Symptoms of cystathionine gamma Lyase deficiency include hypertension due to lack of hydrogen sulfide normally produced from a reaction including cystathionine gamma Lyase along with another enzyme. Hypertension occurs because hydrogen sulfide is a vasodilator and additionally plays a role in regulation of cell growth. Cystathionine urea, or increased cystathionine in the urine, also occurs due to this deficiency. Additionally, cystathionine gamma Lyase deficiency has been seen to play a role in Huntington's disease, a genetic disorder that causes breakdown of brain nerve cells and eventually leads to death. Symptoms of Huntington's disease include changes in personality and cognition and motor abnormalities such as lack of coordination and instability.
The treatments of cystathionine gamma Lyase deficiency are not well developed. There are, however, treatments for some of the symptoms. One of the treatments would be statins, which are pharmaceutical drugs that can be used to decrease hypertension by lowering the cholesterol levels in the blood and therefore allow less buildup of plaque in the arteries and lower the blood pressure. This occurs by inhibiting the HMG-CoA reductase enzyme that makes cholesterol. Another treatment would be to use hydrogen sulfide donors such as sodium hydrosulfide and S-diclofenac to increase the levels of hydrogen sulfide in the body, which have been diminished by the cystathionine gamma lyase deficiency. The role of hydrogen sulfide is a vasorelactant and a regulator of cellular proliferation and/or apoptosis. Lastly, paroxetine treatment or vitamin B6 supplementation can be used to treat nerve cell damage, which is an effect of this deficiency as well.